Thank you.
All right. My slides visible. Thank you so much, Swati. Once again, very good morning and assalamu alaikum to everybody. And I see this attendance is quite good. Aligarh is pretty foggy and it's a bit cold as well. I don't know how is it in Delhi. It's afternoon now. So today, this is not going to be an education lecture, rather it's a concluding one where I would like to summarize some of the stuff that we have learned so far, and also some recommendations to the policymakers and to the universities and to the students and participants here about how to proceed from here. The take-home messages in the 5-day course, what we talked about, what we discussed, what we worked upon, that how do we take it forward. So I would try to do that in a few spots. My last lecture. So if you have any questions or any concerns, I'm available now or later on as well. So just a recap that we started with a new, not new, but I would say a rare concept about using multiomics for finding the therapeutic targets. As I had said in the beginning, that we always knew that, yes, this is something that is relevant, but we didn't know how to do it. So when the genomics and transcriptomics and proteomic technologies started to come up, we started doing it on the high throughput scale. We started to be successful with all the 3 billion bases that were known as the components of DNA. Once it was possible, then yes, we thought, why not we start integrating them? So that's how multiomics is picking up. And still, I told you that there is a limitation on the data analysis part, that we still is struggling to make sense out of all this data that is being generated in terabytes. So I hope you agree with me that cancer problem is like a jigsaw puzzle that we need to put together for finding the right therapeutic target. If we are not doing it, we are missing the picture, and we are not going to improve significantly in terms of the cancer care, in terms of making the life of these cancer patients better. And obviously, we have been more successful in preventing cancer. The early diagnostics, the liquid biopsy-based diagnostics, then the prevention strategies, the work on the lifestyle factors, reducing obesity and reducing other compounding factors. They have all been able to bring down the incidence, but still, once we diagnose and once it's progressive, the cancer still have a dismal problem in terms of treating it. So we must embrace or acknowledge this idea that we should look around the cancer research problem in its entirety and not on just a piece. This is the snapshot. This is a summary figure that I showed you earlier as well, that what we are doing here, we are trying to generate the data from patient samples. We are trying to generate data from cell lines. We are trying to generate the multi-omic data from even the liquid biopsies. And once we have got this data, the genome, epigenome, transcriptome, proteome, metabolome, and there are other omics as well, we put the latest in the data analysis tools, and then we are able to get either biomarker discovery, or we can find the targets, And we can also look into the mechanisms of how these cancer cells are changing and how these cancer cells are uh, shifting their target. The problem of drug resistance is there as well. Once a person is cured, again the cancer comes up, and that is even more lethal because the drugs are no longer working. So that's why we need it for all these applications, and it has to be in a personalized manner and personalized treatment plan. This is how this is like a roadmap that we are trying to follow, and we are trying to develop evidence for every step of this paradigm. Excuse me. So a big help has come from uh, the advancements in the machine learning and artificial intelligence technologies. All of you know that how disruptive it has been. It has changed the way we are doing our work. There's nobody who's not using ChatGPT or Copilot or Gemini. In fact, many of us have used it for preparing for this course and presentations. So this is something that is integrating into our lives, and it is also helping to integrate the multi-omics. And because there's no other way, no human mind can put together billions of data points and find a pattern out of it. It's only the machines that can do it. Yes, we tell the machines how to do it, but we can't do it. So that's as simple as that. And but the real challenge is to find out a use of these information. That how do we use it for patient stratification or patient characterization, or using it for developing biomarkers, or even for the diagnostic purposes in digital pathology. And the quest for finding out the mechanism continues, because what all we are talking about is based on some fundamental basic research that has been done over the decades. We shouldn't forget that this is the backbone of all the science that we are trying to put into the clinic. We are trying to help the patients, but it is the work of decades of basic research where we were able to know the mechanisms of these genes and the cellular pathways, the signaling pathways, or if you talk about the epigenetics, or you talk about the metabolism, or you talk about the drug. There's a lot of basic work that has gone into making the platform where these things are now feasible. You heard from Dr. Asim about different aspects. I think he has a lot of basic stuff covered here, and you learned about how the cancer is induced, what are the pathways involved, and how we can target those pathways. So these are some of the snapshots from his slides that cancer could be a result of multiple mutations, which are accumulating over the time, and then it is uh, reaching to a threshold, which takes the shape of a full-blown cancer. And while these mutations which are actually errors in the DNA, while these errors are happening, there is error correcting machinery found inside our cells. This is the DNA repair machinery, which is also active. So it's a race between the DNA repair machinery and the insults or the errors that are happening in the DNA. Whichever wins causes the effect. If the mutations they win, and doing a mismatch repair or any other repair loses, then we see the accumulation of these mutations. We have, like I talked about the example of a whale. The whale has got a very proficient and very efficient, fast DNA repair system. That's why even such a big animal, and there's a lot of dividing cells there, they don't get cancer. Can you see my screen? I think there was some. Hello. Can you see my screen now? Okay. And is the slides are visible? Let's do that. How about now? Wait a second. All right. Is it okay now?
Yes, yes.
Okay. Can somebody please help me with telling him where did I leave? Where was the interruption? Swati, did you see this slide? Hmm. Anyways, so let me get ahead with this. Oh my God, that was long back here. Okay, so this is something that I was doing a recap for Dr. Asim, that we learned a lot of things about androgen receptor, prostate cancer, and how does RNA DNA repair work, and these mutation progression and mutation accumulation leading to the formation of the cancer. And we also learned about role of hypoxia and thrombosis from Professor Zahid Ashraf. We learned about metabolic aspects from Professor Hamid Ashraf, epigenetics from Dr. Siddiq, and recently, today you heard about preparing the drug libraries from Dr. Afi Anam, which is a very good fit between in the form of concluding this course that we are doing in finding the targets. But once the targets are identified, we definitely need these drug libraries for going to the next level of clinical translation. So we have learned about all these things, and I picked up from this one of our slides, and the last here, the last message here about the precision medicine begins at the molecular level and rational drug design is the driving force. It's a wonderful conclusion because we, even if we find the target by using multi-omics and machine learning and alternative and artificial intelligence, what are you going to do with that unless we have the right molecule, right drug to target it? That's only when it becomes clinical reality. So we need definitely the help of these rational drug design and chemists to make it a clinical reality. Now, how a message for all the participants here that how are we going to do it? It's about collaborating effectively. That we should be able to come out of our boxes and shake hands to collaborate. We leave our insecurities, and for the students, don't think that I am in a particular course, so I cannot talk about other things. No, you should be opening up to collaborate effectively for the researchers, for the scientists, for the doctors. We should also be leaving our comfort zone and at least reaching out to the people from other disciplines who can help us out. There's no point in reinventing the wheel, rather, we should be shaking hands to make things more accelerated. Thinking out of the box, and I think here reading some of the science fiction novels would help because while we are working on a specific problem, it's very difficult to come out of that focus and try something new, but this is definitely needed. And when I started talking about this multi-omics about 10 years ago, people were thinking that I came from Mars. They thought that I have been talking about something very out of the world stuff. But now you can see that the publications are coming in, people are embracing this idea, and it's now even a clinical reality in some countries, in some areas of those countries, hospitals are adopting it for doing multi-omics, longitudinal studies are coming up. where individual patient reports are coming in and they are being recommended based on the multi-omics paradigm. So thinking out of the box would definitely help you in looking something extraordinary and looking into the future. Mentoring is one of the very important aspects. Get mentored because and mentoring is not something that is to be done only for the students. Even when I relocated to India, I looked for mentors who can tell me about the Indian science system, how the funding is happening, how things work here, how collaborations work here. So there's no age or stage for getting mentored. Rather, whenever you feel like you need something that that needs mentoring, find a mentor and get mentored. That's always helpful. Now, since I have to talk about the framework as well, so I would suggest that in our university we need to have more efforts so that we can contribute to the national and international efforts in finding cancer therapy targets and curing cancer. And so we need more efforts in teaching as well as research. And we need more people even to work in cancer biology to contribute to the international arena. We also need more industry academia connect, which is totally missing here. That even if we have got a target, even if we have the drug molecules available, who is going to test them and who is going to commercialize them, who is going to put them for the patients. So we definitely need more industry academia connect, and we also need it more for the funding of the research because now the government funding is really, really difficult. Yesterday only, the Anusandhan National Research Fund, they sent their results for research grant proposals, and there was a bloodbath. There was hardly very few projects that got funded. So we need more industry academia connect to do that. More consortium efforts, and this is what we have started at Aligarh Muslim University. We have made a consortium with the help of 7 different departments. So there's a problem. The problem here is cancer for us. And we are trying to develop a collaborative exercise with the help of different expertise. I think such consortiums are needed in other institutions as well. And if there's any help that we can extend to establish those consortium and help them with the functioning of these consortium, we'll be happy to do that. And we have to push for this concept of precision and personalized medicine. Because so far, I think we are living in that archaic stuff where I see the funding priorities are based on the type of cancer, head and neck and lung and breast. But we are moving from that concept. We are moving towards precision and personalized medicine. We are moving towards not the tissue origin, rather the molecular origin. So we need the funding authorities to acknowledge this concept of precision and personalized medicine, so that we can have better outcomes, better societal benefits, better benefits in terms of the health of the people. So we have to think about it because there's a misconception that it's going to be very expensive. Rather, when you talk about the precision and personalized medicine, it could be more economical because we are not allowing unnecessary tests to be done on a patient. We are not allowing any unnecessary drugs to be given to the patient. We are not allowing any unnecessary surgeries to be done on the patient. If you look into the statistics, a lot of unnecessary work is done on the patients just because it's hit and trial. They don't know. They don't know the target. They don't know the person, and they are wasting their own resources as well as the resources of patients, causing them problems, mental trauma, and other things. So don't think that multi-omics is something very fancy and it cannot be done. It's very expensive. No, it is pretty much doable and it should be done. Unless we do it, we are going to be going in circles and there might be no solution to it. And I'm sure this technological improvements will bring down the cost. I have shown earlier slides that there was $1 million genome sequencing done in 2001. Now that same genome can be sequenced for less than $1,000. So when the technology improves, you can see that in case of cell phones, the electronic gadgets that you're using now. Once they were so expensive, not within the reach of a common person, now everybody has it, everybody can afford it. I believe that once technological improvements will be there, it will bring down the overall cost of precision and personalized. And definitely, since this course is focused on finding the therapeutic targets, we have to think differently. We have to bring a paradigm shift in how we identify targets using multi-omics approach. This is another slide that I had shown you earlier, that we can also use the same approach for developing biomarkers by using the existing data sets and then putting them into the integrated analysis pipeline, validating them using the in vitro or in vivo or ex vivo stuff, and we can get multiple biomarker discovery. Biomarkers are essential for practicing precision and personalized medicine. There is no, it will be very difficult to do precision and personalized medicine without biomarkers. Biomarkers are definitely essential for doing these. So this needs to be developed simultaneously while we are developing the therapeutic target. And keep in mind, the final outcome has to be precision personalized medicine that saves lives. We are not doing just for the sake of publishing papers or doing academic exercises. Rather, we have to keep in mind that we have to save lives. And this is what will give us the passion and to work day and night towards this outcome. With that, I think I've come to my last slide about the acknowledgement. And this acknowledgement is, I'll start with our Vice Chancellor, Honorable Vice Chancellor, Professor Naima Khatoon, and the chief guest that we had. I'm summing up the two GAN courses here, so you'll see the names from previous one as well. We had Syed Ahmed, who was the CEO of Tech Invention, and Professor Zahid University. Dr. Mohammed Muzamil, the principal of Zakir Hussain College of Engineering and Technology, Professor Sar Ahmed, the director of the Nanotechnology Center, and Professor Jahangir Wasi, our national coordinator for the GAN course in AMU. And I would love to thank my co-faculties, Dr. Mohammed Zainuddin from Jubilant Therapeutics, Professor Hamid Ashraf, Professor Dev, and Dr. Afi Anam. And a big thank you to my PhD students, Swati Acharya and Sayyida Shabi Zafar. They have done a lot of work. In fact, they were the backbone for and encouragement for me to do these courses. Without them, it would not have been possible at all. And organizers, Dr. Kasid Anwar, Dr. Sayyid Abdul Ahmed, and Dr. Salih Nassim, they have worked tirelessly to make sure that everything goes well. The previous one was in person. This one is online, and both of them had their own challenges, and both of them had got their own problems. So I would give a big thank you to the organizers, and definitely the peripheral setup by the grant section finance officer. This is all definitely acknowledged, and this is needed to do these courses. Finance officer, PRO, polytechnic staff, learning garden, and everybody who supported. And last but not the least, the main focus and component of this whole program is about the participants and volunteers. So thank you for attending this course. Thank you for registering this course. And I hope you go away with some messages, some passion, some fire in you that can help you in saving lives that are being destroyed due to cancer. With that, I would end my presentation, and I would love to take any question or discussion or I'll leave this. There's something from Padmavati. I have a question regarding successful grant applications. Do you think including meta-analysis data as a preliminary result would increase the success rate of the proposal? I would say this is looking to be the future because a lot of data is available and we don't need much resources to analyze those data. So please go ahead and publish those results. meta-analysis are more powerful than the original research articles because they are built up on multiple publications. So the evidence that comes out of meta-analysis is stronger. But I don't know how the funding authorities take it. Probably they might not consider it as a valuable exercise. But if I'm in the funding authority, I would definitely consider it as a valuable contribution. But the wet lab stuff is more and more difficult because of the funding crunch as of now. So we should be shifting towards data analysis, and that's very, very valuable everywhere. Okay, so let the audience warm up to and ask questions. So we keep the session live until 1 o'clock, and I'll be hanging around here. And then we also have the exam from 3 to 4. And this exam, Swati, can you tell us about the conduct of the exam once again, please? So at 3 o'clock, we'll only the participants will sign in with their registered email ID. Yes, and there will be 100 questions out of 115 that needs to be answered. Yes, and all these questions are from either my slides or Professor Dr. Asim's slide. So the questions have been prepared from only these two sources. They're pretty simple and it's a good exercise. And if you want to take the credit for the course, the exam is mandatory. If you don't want it, then yes, you can skip. Okay, so I think Padmavati, you are talking about the I want to write a project proposal based on the omics studies. What are the factors should I consider to get a successful funding? So the best factor is to tie up with somebody. If you are a computational biologist yourself, it's fine. If not, then connect with, make a good team with some computational biologists. So that will be one thing. Then identifying the problem, and if you can use some in-house resources like the Indian genome that DBT has recently released. If you want to work on that, that will be even better than using the data from TCGA or Caucasian population or other population. If you can get your hands on some in-house data, that will be even better. If you have got data from the university, from your own place, that will be more robust. There are specific calls by DBD. Now they have a section on computational biologies. Earlier they did not have that. So you can apply under those sections. They invite grants twice a year. So you can apply under that section, which is totally data driven, and it should be easier than putting it for the wet lab studies for genetic data. I hope that answers your question. And you can connect later on as well for more details about particular funding agency that needs to be tapped in for getting the fund. Yusra is asking what is the exam passing cutoff? If we just give me a minute, I'll tell you about the grade. So the exam passing cutoff is 35, and I've already shared this document which will give you all the details. So if you can see in the chat box. Yusra. think, Swati, we are getting no more questions. So we can close the session here and we reconvene at 2:45. Reminder email will be helpful. If we send a reminder email to all of them, the 2:45, everybody should log in so that we can start the exam at 3 p.m.